Hello, everybody. Welcome to Micro 4100 General Virology. So, my name is Dr. Ali Jazire, and I will be your instructor for this course. My email address is ajazire@calstatela.edu. The class meets for lecture on Monday, Wednesday, Fridays at 8 to 8:50, and then I have office hours on Monday, Wednesday, Friday, 9:15 to 9:45. Uh, Please send me an email or a message in Canvas so we can schedule the office hours. Uh, there are some prerequisites for this course, and these include a very good understanding of cell and molecular biology, as well as having completed courses in microbial genetics or general genetics and general microbiology. So please ensure that you have the prerequisites fulfilled in order to be able to succeed in this course. The outline of the course is basically we have lectures and class discussions. There are chapter readings. There will be case studies that students will be working in groups and present the cases in the form of a PowerPoint slide presentation, as well as providing a written report. And also, there will be 3 examinations. These 3 exams, each worth 100 points, the case study worth 100 points, so total of 400 points. The course material are all available on Canvas, so you can access the course material there. And the expectation is by now you have familiarized yourself with the structure of the various modules of the course on Canvas course homepage. If you have not done so, please make sure to do it. The textbook should also be on reserve in the library. So, what we plan to accomplish during the course is to discuss the biology of the viruses, their replication, diseases caused by viruses, how viral diseases are being transmitted, possibly prevented, and possibly treated. So, we'll talk about the transmission, prevention, and treatment of diseases caused by viral infections. We will also discuss host cell response to viral infections. What would be the body's normal response when a virus has successfully been able to cause an infection? We'll briefly touch on the role of viruses in biology. And uh, as an upper division course, it is imperative for the students to be able to solve problems. Solving problems will lead to new knowledge. So don't expect the exam questions to be just directly from a slide that you see, you memorize it, and then you can take the exam. No, it's basically connecting the dots, if you wish, and being able to critically think. So, hopefully, throughout the semester, by trying to connecting the dots, we will work on developing and strengthening those critical thinking skills that we all have. The required textbook is called Basic Virology, third edition. There are some chapter readings, some chapter questions, and you can get the book from bookstore or student bookmart or online, such as Amazon. As I mentioned, it's on the reserve loan at the library. There is a second reference book called Medical Microbiology. It's optional. If you want, you can borrow it, look it up. We basically try to refer to this book for the case studies, for both the oral and written reports of the case studies, but you are fine uh, to search for other resources online as well. There are some online resources. Textbook publisher is the link for the textbook publisher here, Blackwell Publishing. And how we evaluate your learning by the written examinations. There are three written examinations, and each worth 100 points. So 300 points total. Then participation in class discussion, regular and active participation in class discussions, and the questions that are being posed. I just want to ensure everybody that you need to participate, strongly encouraged, but I want to ensure everybody that it's not that if you feel you do not know the correct answer to a question, you just be quiet and not participate in the discussion. That should not prevent you from participation, because if you knew the answer to all of the questions, you would have not been here and you would have not been taking this course. Throughout the semester, we are trying to learn from each other. So please do not be shy and try to participate. The format of the exams could be multiple choice, true false statements, identifications, maybe short essay type questions and long essay questions. Uh, I will put you guys in groups of 3 or 4 for case studies, and then uh, the case study questions are already being posted on the Canvas course homepage. You can refer to that, familiarize yourself. There are basically a series of questions. A case is being presented, followed by a series of questions. You need to address those questions and also provide a presentation on it and a written report. Oral presentation part of your case study is worth 50 points, and the written report is also worth 50 points, total of 100 points, plus the 300 points of the written exams would be 400 total. Total points are 400, and it's broken down to, for example, 360 to 400 is A, 320 to 359 B, 280 to 319 C, etc. More detailed description can be found on the course syllabus. For those individuals who actively participate in class discussions, there is up to 20 extra points. It doesn't mean that you just discuss an issue once and you expect to get 20 points and that's it for the rest of the semester. No, it is active and regular participation. One thing that is of extreme importance is the academic honesty. The academic dishonesty can be in the form of cheating, which is deceit, trickery, artifice, or plagiarism when you steal someone else's ideas and path of the ideas or words of others as one's own. So, cheating and plagiarism, whether it's in your writing the reports or during the examinations, not only violate the spirit of learning, but also constitutes academic dishonesty. And this is the one thing that I am very sensitive about and very unforgiving. I have included a specific module on Canvas course homepage labeled as academic honesty. There are many articles there. Familiarize yourself with academic honesty and the ramifications of not following it. And you can also refer to the link provided, calstatela.edu /judicial affairs. So, why do we even bother learning? Why do we even bother learning virology? Or how do we do that? Let's talk about what should we do to learn this course. This is a very difficult course. Unlike bacteriology or microbiology that you have 1000 level, 2000 level, 3000 levels of it, and then you get to the 4000 level. Virology starts as a 4000 level course. So you have a lot of concepts are new, a lot of the terminology are the first time you're hearing it. Therefore, we need commitment. Students, I would strongly recommend them to attend the lectures regularly and participate in class discussions. If you miss a class, it is the student's sole responsibility to catch up with the material and inquire from their peers what was done in class, where we are in class, and catch up. And at the minimum, you need to spend 2 to 3 hours a day in addition to the class time for study before you realize material will accumulate and it will be very difficult to get around all of them. Try to read and study assigned chapters in the textbook and focus on topics that are covered in the lecture. So, not everything in the chapter textbooks are what we cover in the lecture, so please make sure that you go over that. There are some chapter questions that can evaluate learning. This is supported by the publishing company. Some of them are similar to exam essay type questions, and you also need to study those case studies. Make sure to understand them. These will be the case studies are a very excellent tool for students to comprehend detailed information about a particular virus and the diseases it causes, how we can manage, control, prevent, or treat it. So now the question is, why do we even study virology? Because virology and viruses affect every aspect of a human's life. We will discuss all of these different scenarios in subsequent lectures. So let's start by discussing some historical introduction by talking about smallpox virus. Before we discuss the smallpox, there is a quote from Albert Einstein that says, "We are able to see so far because we stand on the shoulder of giants." I believe what he's referring to giants are those selfless, humble scientists who have dedicated their entire life to humanity and have uh, been trying to solve scientific and life puzzles. So, in the 10th century BC, smallpox was developed. It showed records of epidemics in Asia of a highly infectious illness that was usually fatal, about 15 to 45% mortality rate or fatality or death. Smallpox is caused by a virus called variola virus. It causes spots or vesicular rashes, as we can see in the slide. And Chinese prevented this infection by self inoculation of the dried pus that were obtained from these lesions of recovering patients. So there were patients that were recovering from smallpox. They had lesions or these vesicular rashes on their body, and the Chinese got took the dried pus from this and self inoculated. Unfortunately, it sometimes did not cause protection but induced disease or even led to the death of the individuals. This whole process was termed variolation, variolation or altered virus of infected patients are being used to elicit or trigger or mount a protective immune response because the body recognizes the pus as a foreign object and it serves as an antigen. The immune system recognizes the antigen, and plasma cells or terminally differentiated B cells will secrete antibodies or immunoglobulins against it to confer immunity. Smallpox caused by variola virus spreads worldwide. There were reports of the disease in Asia, Africa, Europe, and America. Let's take a look at the smallpox in the 17th century. Dr. Edward Jenner from England is the person who has done a lot of studies on smallpox, and he noticed uh, a similar pox infection in cows. And what he observed was that the milkmaids that are infected by cowpox are protected against smallpox. So those individuals who got cowpox were resistant or developed an immunity to smallpox. And Dr. Jenner developed the first vaccine where he used pus from cowpox lesion obtained from milkmaids to inoculate into the patient or at risk individuals to prevent smallpox. So, this sort of vaccine that Dr. Jenner had discovered prevents infection and also does not cause diseases. And this procedure was termed vaccination, coming from the Latin word vaccinus, meaning cow, which is basically the application of a less virulent virus, in this instance, cowpox, to elicit protective immunity or to develop an effective immune response against smallpox. So, if you have a less virulent virus that is used to elicit protective immunity, the term is called vaccination. What about research on smallpox in the 20th century? In the 20th century, the scientists have used chick embryo, as shown in the upper left uh, corner slide picture in the slide. They've used chick embryo and cell culture as a host to study virus in the lab. Unlike bacteria that we can grow them in culture and we can study them, viruses definitely need a host to be studied. Viruses are inert microorganisms. They look like dead organisms on the outside of the body, but once they get into a host, they start replicating, expanding, and causing diseases. So this variola virus is a complex virus and it replicates or makes more of itself in the cytoplasm of the host cell. So virus needs to get into the host cell, get into the cytoplasm of the host cell and starts replicating, making more of itself. And this is basically the basis of R&D or research and development for biological warfare back in the day in the United States and Soviet Union. So eradication of smallpox in the 20th century. In 1967, World Health Organization or WHO initiated a program to vaccinate susceptible persons during an outbreak, and this will stop the spread of the virus. In 1977, there was the last natural case of smallpox in Bangladesh. In 1979, World Health Organization declared smallpox is being eradicated, and this was the first success of eliminating an infectious disease agent in human population. Moving to the 21st century, scientists have discovered that there are some benefits to the smallpox virus. So, in a sense, they consider it as a friend. Basically, they get a plasmid vector, as shown here, as uh, the plasmid vector contains vaccinia virus gene. They remove part of the gene and insert a foreign gene. For example, the gene that encodes for HIV GP120. Then they would infect host cells with both a wild type and a plasmid wild type vaccinia virus and a plasmid vector. So what will happen? Virus gets into the cytoplasm of the host cell, starts replicating, making more of itself, and during this process, there would be the recombination of wild type vaccinia virus DNA and recombinant vaccinia virus gene, where it was in the plasmid vector. So the end result is through the recombination events, we have a novel recombinant vaccinia virus that contains the foreign gene as well as the wild type original vaccinia virus. And this recombinant vaccinia that has the vaccinia virus gene as well as the GP120 gene is being used as an experimental live recombinant vaccine to elicit immunity against, in this instance, HIV GP120, or in general, whatever foreign gene that we include in the plasmid. It was also in the 21st century that scientists realized that smallpox, uh, in addition to its utilization in recombinant DNA technology, can also be a foe or an enemy. It creates a lot of fear due to the fact that it can cause deadly epidemics, and unfortunately, it can be used in bioterrorism. Let's talk about another virus called tobacco mosaic virus, TMV, that was discovered in the 19th and 20th century. Scientist Ivanovsky from Russia noticed that a bacteria free filtrate. So basically, he had these tiny filters that bacteria could not pass through, but he noticed that this bacteria free filtrate of the plant's sap still remain infectious. So the initial thought was that, oh, maybe there are some small microorganisms, and uh, Stanley from United States was the one who crystallized TMV from plant extract, and the question remained whether this crystallized TMV, is it a macromolecule or it's a non-living organism. And TMV is the first virus that was visualized. Electron microscope showed a small rod-shaped helical particle. So it looks like a bacillus, if you wish, but it is a small particle, and the size was in the nanometer range, which is 10 to the power minus 9. In 20th century, scientists also discovered bacteria viruses, those viruses that can infect bacteria. So Two scientists, Twort and the Herald, discovered that bacterial colonies become clear, water and watery, and the bacteria will be killed. So, if you have a long growth of bacteria, as you have learned in microbiology courses, if there is viral infection, the area that viruses have successfully infected the host cell, in this instance the bacteria, it becomes clear, and the virus, or so-called a plaque, and that's called plaque assay. We'll talk about it later. So the scientists named the viruses that use bacteria as their host as bacteriophages. So, how they do this bacteria virus research? Two genetic a physicist and a geneticist collaborated on genetic studies of T4 phage. They used, as I just mentioned, plaque assay, where, as you can see in this picture, wherever there is bacteria and virus has successfully infected the bacteria, the colonies become clear because the bacteria has died. And this was basically the beginning of molecular biology. And Loaf studied lambda phage in Escherichia coli, or E. coli, and he saw different things. Sometimes the infection resulted in no cell lysis. He also noticed that the virus genome gets incorporated into E. coli's DNA, and sometimes there was no viral replication. This is termed lysogenic or latent infection. In the 20th century, scientists also discovered RNA tumor viruses. Ross from United States discovered a viral infection of chickens that results in a type of cancer called sarcoma. Related viruses also shown to cause leukemia in cat, mouse, and cow. These are unique, and there are also unique viruses called retroviruses, that they have an RNA genome that can be converted to DNA, and viral DNA gets integrated into host cell DNA. And these viruses can also be called foamy virus because they're common in many animals, but they do not cause any diseases in animals. In the 20th century, similar to RNA tumor viruses, Shope from the United States discovered that a DNA virus can cause papilloma, or the type of cancer, in rabbits. And this virus infection changes the host cell dynamics. There are all these signaling pathways, cell cycle regulations in the host that, when the virus infects the host, will change those pathways and results in cancer. Other examples of the DNA tumor virus research. There were other viruses related, the DNA viruses, in mouse, cow, horse, primates, and humans. And scientists were able to identify viral proteins in tumors of the animal. And this is a very important tool for cancer research. And this the ability of the virus to cause these disease or cancer is and the interaction between the host and the virus, it's a very good research tool to study cell regulation. Emergence of virus pathogens. Certain human infecting viruses, certain viruses that cause infections in human are known to be able to persist in the wild. Examples are rabies virus and West Nile virus. The persistence of these viruses in the wild requires a broad host range. In other words, they need to have multiple hosts in the wild so they can survive. And the question is, how does a seemingly new virus emerge to sicken humans? Let's talk about emerging viruses. Viruses emerge because there may be a change in the balance between the host and parasites. Some of the of possible factors that play in favor of the virus are the genetic, environment, political issues, social issues, international travel. Uh, Emerging viruses can also be as a result of human contact with wildlife, such as HIV, SARS, coronavirus. Emerging emerging viruses can also be as variants of endemic milder pathogens. Example would be avian influenza strain H7N9 in birds. And this slide, Emerging Viruses 21st Century, shows that in the 21st centuries, in different regions of the world, different viruses were emerged. So let's talk about some of these emerging viruses. The very first emerging virus is Ebola virus. It causes fatal hemorrhagic fever. And the host reservoir of Ebola virus is unknown, and human is an accidental host. It's not the main host. There's another emerging virus is HIV, human immunodeficiency virus, that causes a chronic infection characterized by immune deficiency. Another emerging virus is a hantavirus or sin nombre virus that causes hemorrhagic fever plus pulmonary infection. Prions are subviral agents. These are proteinous infectious particles. These are subviral. Prions are not viruses, these are proteins that cause subacute spongiform encephalopathy. We'll talk about prions. There's a lecture dedicated to prions, actually. Other emerging viruses include West Nile virus, which is an arbovirus, meaning that it's an arthropod borne virus. Most of the infections are asymptomatic, and in 1% of the cases, it can cause encephalitis, inflammation, and infection of the brain. SARS CoV, severe acute respiratory syndrome, coronavirus, which is it often causes fatal pneumonia and the reservoir in wild cats. Emerging viruses also include influenza A, that has numerous animal hosts. It's a very highly infectious virus that causes pandemics, and the reason for uh, it being highly infectious is due to the ability of the virus to rapidly changes its antigens so it can go undetected by the immune system. Avian influenza virus is H5N1. This is the serotype, and it rarely causes human infection, but unfortunately, if it causes, it would be fatal pneumonia. There's a novel swine flu like virus that is H1N1. So let's talk about the top 5 reasons why we should get flu vaccine. Flu is a serious illness, a serious viral infection. It can lead to secondary bacterial infections, pneumonia, and death. So, vaccine is the best way to protect ourselves. Vaccines are very safe because they are killed viruses and they cannot give you flu. Vaccines avoid spreading flu to others, in particular high risk people, such as those who are very young or very old, will be severely affected by flu. And lastly, flu vaccines are available at no to low cost. You can go to student health center and receive your vaccine. Other emerging viruses include MERS, CoV or Middle East Respiratory Syndrome coronavirus that was identified in 2012 in Saudi Arabia, causes severe acute respiratory illness, and unfortunately its mortality rate is very high, one in two individuals, or about 50%. MERS-CoV is slightly different from human coronavirus, and it's more similar to the virus that is found in bats. Viral virus ecology. Viruses naturally exist within host organisms in any ecosystem, and their presence or their coexistence with their host is significant for plants, animals, and human health. Ecologists consider viruses as being living entities, and they theorize that viruses play roles that are comparable to cellular organisms. Example: a predator or virus can be a parasite. Many viruses interact positively with hosts by expressing host genes and by protecting hosts from other organisms. Viruses play significant roles in ecosystem. They have important roles in all ecosystems. Uh, they can limit host population density without extinction of the host, and also participate in population decline and resurgence. Viruses also select for host diversity, where they prevent dominance of any one species. So there will basically, it won't be an overpopulation or overgrowth of one species. And viruses also foster evolution of many distinct host species. Viruses also play significant roles in marine ecosystems. For example, when marine algae overgrow, viruses can control algal bloom or their growth. Uh, Viruses have the highest number and the most genetically diverse forms of life in oceans. Viruses also, um, on a global scale, they play very significant role in carbon balance. And let's talk a little bit about mutualism of polynaviruses. Uh, in natural ecosystems, acute viruses, when the virus acutely infects an organism, it kills the host cell. Virus enters the host cell, goes to the specific place and starts causing the disease and kill. However, these acute viruses can be outnumbered by persistent viruses who come along for the ride. These persistent viruses that are just there may evolve traits that basically confer positive benefits in the virus and host relationship. Example would be parasitoid wasp that lay eggs, then virus in insect larvae, and once the virus gets into the insect larva, they express viral proteins in larva. These viral proteins protect wasp eggs from host defense that usually encapsulate and kills it. So, under normal circumstances, if a wasp lays egg, host cell can control it. Basically, encapsulates it and gets rid of it. But if there is a viral infection, virus can protect this wasp egg uh, against host defense mechanisms. In 1959, the question was raised, what is a virus? Some people thought, oh, you know what, a virus is a virus. Whether or not viruses should be regarded as organisms is a matter of taste. And this comes from a French Nobel laureate, Andre Wolff, uh, that basically leaves it to the judgment of the people who are studying it. However, in 93, another Nobel laureate, Joshua Lederberg, said that the very essence of the virus, its fundamental entanglement with the genetic and metabolic machinery of the host. And in 2020, there was a little bit more information about genome. So, uh, Greg Gibson from Georgia Tech said that how a clash between our genes and human life is making us sick. Basically, they're referring to the genetic elements within the viruses. In the 21st centuries, there was a more complete definition of the virus being proposed, and that is viruses are very small agents that can pass through filters less than zero point 0.2 micrometer. They are non-cellular organisms, so by themselves they do not have metabolism. They have a simple composition. Their genome is either DNA or RNA, protein, and sometimes there would be glycoproteins in their lipid membranes. From a biological standpoint, viruses replicate by assembly of preformed components. These components are nucleic acid and various proteins, and not by host cell division. And viruses are involved in our lives. The origin of the virus is now the question is by studying a phylogenetic tree which shows the degree of relatedness between various organisms. As you know, we have three kingdoms: eukaryotes, eubacteria, and archaea. And by genetic analysis of viruses and looking at phylogenetic trees, they wanted to see whether there is a degree of relatedness between virus and these three super kingdoms. And it appears that there is an ancient association of virus with host cell. Viruses most likely do not come from host cell because they do not have any genes for ribosomes and metabolism. DNA polymerase gene uh, was another tool to study the origin of viruses, and it was found that viral enzymes are more similar to their ancestral forms and viruses' origin of DNA based genetics. So that was it's because of their ease of use in modern molecular biology and DNA based genetics. Viruses can be considered as one of the important tools in uh, genetic studies. Uh, Others have a different view of the viruses. They want to see whether there is a cellular origin for viruses. For example, the complexity of variola or smallpox virus, which it has a large double-stranded genome. It has distinct brick-shaped structures that is large size, 400 nanometer, has double membrane like host cell plasma membrane. It has lateral bodies. Its replication happens in cytoplasm using both viral DNA and RNA polymerase, and they resemble cell organelles. So, all of these will point to the fact that viruses may also have a cellular origin. Lecture summary: smallpox was one of the ancient plagues that killed a large portion of the infected population. Vaccination came from understanding a weakened or altered virus stimulates our protective immunity, and there is no reason to for us to fear smallpox virus any longer. And as a matter of fact, smallpox virus has become an important research tool. The study of tumor viruses, DNA and RNA tumor viruses, has helped us to understand the cellular regulatory mechanisms and the cancer process. An emerging virus appears when there is a change in its host and parasite balance. Viruses are biological and important part of life. And overall, we do not know the origin of viruses. Reading for assignment for this is from chapter 1 of the textbook. Thank you so much for your attention, and this is the end of the lecture on introduction to viruses.